Hello, in this video we will discuss about the fermentation. So the aerobic fermentation and anaerobic fermentation, and there basically we will understand the benefit of the fermentation and what is this. So first of all we should need to understand the definition. The fermentation is a metabolic process that produces the chemical changes in organic substrates through the action of enzyme. For example, the glucose will convert into the alcohol due to the enzymatic activity in the Saccharomyces or lactobacillus. So anyhow, let's begin to understand the what is the fermentation including organisms. So here is the lactobacillus bacteria, Aspergillus and Saccharomyces. So the Saccharomyces is also known as the yeast. So remember about that. So the yeast is the Saccharomyces. So Saccharomyces basically. So in this way, all living organisms contain glycolysis. So first of all, we should need to understand what is the glycolysis. Glycolysis here is a sugar, the half glucose which that convert into the pyruvate 3 carbon containing molecule due to the enzymatic activity without oxygen consumption. So here the glycolysis NAD reductase enzyme lead to NADH formation and the phosphoenol pyruvate formation, and the pyruvate kinase which that is converted by the also produce the ATP to produce pyruvate. This is the basically summary of the glycolysis. While here is the intermediate molecule is the pyruvate which that convert into the acetyl CoA if the aerobic condition. While on the other hand, it is also converted into the pyruvic acid by the NADH usage to donate proton. So the NADH dehydrogenase enzyme used for it, and it can be reversible. Anyhow, let's begin to understand the Aspergillus. This Aspergillus is used for the fermentation of soy sauce, and the lactobacillus. Lactobacillus is used for the, this is the bacteria used for the cheese and yogurt production. So this is the fermentation process which that is a lactic acid will be formed due to the fermentation rather than convert into the alcohol, while the aspergillus convert into the soy sauce. And here is the Saccharomyces. Saccharomyces with in the absence of the oxygen, so it will convert here is the soy sauce from the aspergillus, and lactobacillus will produce the cheese and yogurt. So the Saccharomyces, if the ethanol and carbon dioxide will produce from the beer. So the beer fermentation will due to the barley and wheat. While on the other hand, the ethanol will produce the red wine and wine, which that is due to the ethanol production from the Saccharomyces yeast. So on the other hand, the carbon dioxide, which that is produced, will lead to fermentation of bread making process. So anyway, let's begin to understand the major mechanism within the cell and what happen in the cell. So what is the mechanism? So remember, this is an anaerobic condition. So the glucose are present in the medium. So this yeast is the unicellular eukaryote, and anaerobes grow on non-oxygenated place. So non-oxygenated place is very important. So this is the extracellular and intracellular place. So this is the glucose will enter into the inside of the intracellular. After this entry of the glucose through transporter channel, it will convert due to the phosphoglucokinase enzyme to glucose 6-phosphate to convert the phosphofructokinase to convert the glucose 6-phosphate into the fructose 6-phosphate. In this way, the ATP will be served. So this is the same mechanism, the glycolysis. So remember, this is the cytoplasm, and in this way here is the glucose 6-phosphate, fructose 6-phosphate, and fructose 1,6-bisphosphate or diphosphate will be formed. After this, it will split into two molecules, dihydroxyacetone phosphate or glyceraldehyde 3-phosphate. Basically the same mechanism, the glycolysis. I am condensed it. I am condensing it to understand just for overview. So this is, I am missing the many reaction, but you can understand here is the 1,3 diphosphoglycerate will be formed due to the NAD will reduce into the NADH due to the NADH reductase enzyme, NAD reductase enzyme. And in this way the ATP will be used to produce ATP to produce the 3-phosphoglycerate, and 3-phosphoglycerate will phosphoglycerate mutase enzyme used to 2-phosphoglycerate formation, and the phosphoenol pyruvate to convert the pyruvate by this mechanism. And this pyruvate will convert into the acetaldehyde by the removing of the carbon dioxide into the extracellular region, and this is the gas will evaporate. While on the other hand, here is the acetaldehyde dehydrogenase enzyme used to produce ethanol by the using of NADH to convert the dehydrogenation of the NADH. So in this way, here is the ethanol will produce, and the carbon dioxide is used for the ethanol beer production as well as ethanol produced through 2 ethanol produced from this. So this is the function, and this is the anaerobic fermentation. But let's begin to understand the aerobic fermentation. So the aerobic fermentation means the glucose or ethanol which that is used within the aerobic condition. So this is the acetobacters which that can be enter into the cytoplasm. When enter into the cytoplasm, so this is the ethanol will entry or live in the outside of the environment from the basically acetobacter. So here is the PQQADH enzyme which that alcohol convert into the acetaldehyde by this enzyme. This is the extracellular enzyme, not intracellular. And this acetaldehyde will convert into the acetic acid through PQQALDH and MCDALDH. So MCDALdehyde dehydrogenase and the PQQALdehyde dehydrogenase enzyme use acetaldehyde dehydrogenase enzyme used to produce acetic acid. And this will donate the electron to the ubiquinone to convert the ubiquinol and to remove the proton outside. And in this way, here you can see this is the ethanol. This ethanol will convert into the acetaldehyde intracellularly if so the NAD acetaldehyde dehydrogenase enzyme used to produce acetaldehyde, while here is the NADH is a dehydrogenase enzyme used to fix the ubiquinone to convert the ubiquinol. And in this way, here is the acetaldehyde will convert into the NADaldehyde, and the NADaldehyde dehydrogenase enzyme used to produce acetic acid. And this acetic acid is the intracellularly can be converted into the acetyl-CoA by the acetyl-CoA synthase enzyme to go into the Krebs cycle to produce ATP, NADPH and FADH2. This NADH dehydrogenase enzyme used to remove the proton to move out through a ubiquinone oxidase enzyme. But in this case, the ubiquinol will donate the proton to move out. So the outflux of the proton will again convert into the ubiquinone. And in this way, here is you can see the 4 proton required for the influx of the proton through ATP synthetase enzyme to produce 1 ATP. So this is the ATP synthase enzyme. And this proton again used to produce NADH and FADH2. So anyhow, in this way, here is the acetic acid. Here is produced extracellularly and intracellularly. That is known as the aerobic fermentation will produce the apple cider vinegar and other vinegar. In this way, the apple cider vinegar will produce, because this apple cider vinegar contain acetic acid. So that is the aerobic condition will require to convert the ethanol into the acetic acid. While on the other hand, the anaerobic fermentation, the ethanol and carbon dioxide, soy sauce and cheese and yogurt will produce. Yogurt is basically is not yogurt is also produced in aerobic conditions. So thanks for watching. Bye.